For those that weren't here, Gary Lang from the Western panel chairing this session on canola and oats. And it's my pleasure now to introduce Andrew Heinrich. So Andrew's has a working career in agronomy across the south coast of WA, 14 years in Esperance. Did some time sounds like a prison term, in the GRDC office, where probably I've got to know Andrew more, and has strong interest in canola R&D. And Andrew's paper is about agronomic and financial outcomes associated with retaining seed from both spring and winter canola hybrids. Thank you, Andrew.
Thanks, Gary. Afternoon, ladies and gentlemen. Thank you very much for coming along to listen to my presentation on the research results from that we conducted last year on our canola program through PAC Seeds. So, as the title says, it's the agronomic and financial outcomes from retaining seed from both hybrid canolas, both winter and spring types. So, yeah, last year, PAC Seeds, we developed probably the largest set of data on generation comparisons in Australia and potentially the world for with the number of trials that we ran. Most of the trials are done, or most of the projects that have been done in the past have been on 3 or 4 sets of data from around the countryside. So interestingly, the image in the background there is from our Yarrawonga site in Victoria last year. It would be the largest trial site that we've had in Australia and potentially around the world for Advanta or PAC Seeds. We had some 5,000 plots, research plots there at that one site. And so, yeah, we're investing good money, or a lot of money into research and development. Now with the hybrids and F1, so the F1 hybrid canola seed is produced from 2 distinctly different male and female lines. So the seed that they produce are all the same because they're a combination of the male and female line. The retained seed that you keep in the F2 or the F3 generations are no longer hybrids. They're open pollinating, segregating populations that will segregate for a whole range of traits, from everything from oil content to yield to height to flowering, you name it, it'll keep on, it'll segregate. And so seed retained from a hybrid that has glyphosate tolerance will also segregate for that trait. And so you've got to expect to lose a percentage of plants when you apply glyphosate to those subsequent generations. So the aim of the work that we did last year was to look at the agronomic and financial outcomes from the retained seed for both winters and spring canolas. So in the spring canolas, we compared fresh F1 seed to a blend of F1, F2 and straight F2 seed. And then in the winter canolas, we compared the 3 generations: fresh hybrid F1 seed, retained F2 and retained F3. So we had 18 different spring type or locations dotted around the country, from east to west and north to south. And as a result of that, we had a wide range of environmental conditions, whether it be east, west or north, south. Last year in WA, we had extreme drought in the north to extreme wet in the south, and a range of soil types right across the country. So that range of environments only adds to the strength of the set of data that we collected. In the winters, we had the 3 sites, one in South Australia, one in Victoria and one in New South Wales, where we compared the 3 different generations of the fresh F1, F2 and F3. I'd like to acknowledge my fellow staff members from PAC Seeds that have helped out with these trials, and also the service providers who manage the trials right across Australia, because without their help, these trials wouldn't have been done and we wouldn't have this set of data. So it's a pretty extensive list of trials there. Please don't try and read it all. But just to give you a location of where all the trials were located and a bit of a rundown. So sowing dates for the winter varieties range from the 19th of March to the 29th of March. The sowing dates for the spring types range from the 19th of June, 19th of June, that'll be a bit late, wouldn't it? 19th of April to the 2nd of June. Growing season rainfall range from 60 millimetres to 435 millimetres, and trial mean yields range from 0.3 of a tonne in the droughted situations to 4.35 tonne. So we had quite a range of environments and results that we were looking at. So drilling down into the spring types. Here we're looking at the emergence of the 6 different hybrids that we tested across the 18 environments. The F2 seeds were all graded to greater than 1.8 millimetres before we packed them for sowing. The F1, F2 blend was created by generating a homogeneous mix of the equal number of F1 seed and F2 before they were packaged. Now we packaged the seed to target 40 plants per square metre, and looking at the springs here, we averaged 65% of targeted population, which is well within the range of between 30 and 100% that have been reported by other people researching canola establishment. So within the F1, we're have some significant variation or some significant differences in establishment between the different varieties, which can be expected because we've got different seed lots from different production paddocks and all that sort of thing, different varieties. So that's not to be unexpected. If we look at the different generations, there wasn't any significant differences between the generations in the F1, F2 blend or the F2, but there was a significant difference in individual varieties between F1 and the retained seed generations. So just a quick summary on seed establishment. F1 seed maximised your plant establishment, and there was significant differences between F1, F2 blend and the F1 treatment and the F1 and F2 treatment, but there was no difference between the blended seed and the straight F2. Moving on to the grain yields. So please, again, it's quite a complex little graph, but it shows very clear and distinct trends that the F1 seed in the pink for each variety across the 18 locations exceeded the yield of either the F1, F2 blend or the F2 by itself. Now, it's very difficult for you guys to see the level of significance, so it's easier to look at a heat map of the levels of significance between differences in generations. And just for those who are not familiar, how to read these signs, and apologies to people over this side, I'll put the pointer up on this screen. So basically, through the middle of the screen, it's a reflection from the top left-hand half is a reflection of the bottom right-hand half. So you have each treatment is listed, pushed the wrong button. Each treatment is listed on both axes, and hence the reflection of where they interact. So when we go and look at the whole heat map again, you can see that the interaction between, or the level of differences between the different treatments is highly significant for each variety across the 18 sites, with F1 being the highest yielding variety or highest yielding treatment of those three. So people will still go and ask, well, what happened in my backyard? Because that's more important than the average. Well, technically that's not correct, because what happens in your backyard in 1 year is a reflection of that one season, one set of management conditions, that single site. So where you're much better off looking at a MET result, so a multi-environment trial analysis, to see how robust that treatment was. So again, very complex map graphic, and so it's much easier to look at a heat map of the level of significance of the differences. And again, you'll notice there's a predominance of the dark and pink salmon in the squares, which indicates that there are highly significant differences between the majority of those treatments. So when you break it down on an environmental, on a local scene, looking at the percentage of variance that can be attributed to each component within the analysis, and very strongly, environment is the driver of differences in yield between environments. The degeneration is the next most significant level, but nowhere near as important as environment. So your local environment is driving the yield potential at that site, followed by your generation, which generation you're choosing to sow. And genetics is a minor contributor across the 18 sites. So breaking down again to the individual sites is to see what portion of variance can be attributed to the different components. So what we've got here is listed as all the sites across the horizontal axis, just ranked on site mean yield. The orange line at the top is genetics, is your generation, so whether it be F1, F2 blend, and the difference attributed to in yield is attributed to the generation, is the orange line on top, and the contribution that genetics or actual variety is the grey line at the bottom. Now, interestingly, there's only 2 sites there where the variance attributed to genetics is greater than is attributed to the generations, and that's the 2 sites at Mullewa and Malali, and both of those sites were extreme drought and very low yielding sites. The other 2 sites that are where it's the difference is in yield is attributed to genetics is similar to that of the generation is Karoo, another extremely droughted site. I think yielded just under 0.3 per tonne. The other site was Cowra, which was a high yielding site, but it was extremely wet just prior to harvest, and another challenging environment to grow canola in. So summarising the information that's on this graphic is that generation is driving the differences in the yield in the treatments on an individual site basis at the majority of sites where you've got reasonable growing conditions. So what does it all mean for a grower? So what we've done here is we've calculated a simplified gross margin. So we've just taken grain yield, multiplied by a nominal grain value, and subtracted the cost of the seed. We've assumed that all other inputs at each individual site was equal, which they were, and that each site was managed to its genetic or its seasonal potential yield. So the inputs of fertiliser, etc., will vary between sites, and makes it very difficult to plot out a full gross margin site by site, hence the simplified gross margin that we're presenting here. Now we're looking at Hyola Blazer TT with the 3 generations. So we fitted a linear regression through each set of treatments, and very interestingly, the R-square return for all 3 was a very high level at above 0.96, and at no point does the regression for the F1, F2 blend or the F2 treatment equal or exceed that of F1. So what that is saying is that your maximum dollar return is driven from your F1 versus the over your retained seed. So that's Blazer. We did a similar thing for a couple other representative varieties. So Hyola Defender CT, which is another non-GM stacked herbicide technology, and we get very similar results to that of Blazer. We have F1 is the highest simplified gross margin, followed by the F1, F2 blend, with the lowest one being F2. Interestingly, the same thing shows up here as on the Blazer. The Blazer simplified gross margin was that at higher yield potentials, so at the right-hand end, the actual dollar value of growing F1 seed to the farmer increases at higher yield potential. So it means more returns to the grower. Finally, the final simplified gross margin we'll look at here is Regiment XC. So it's a GM stacked herbicide technology. And again, at no point does the F1, F2 blend or the F2 simplified gross margin exceed that of the F1 fresh seed. So just a quick summary for the spring types. F1 hybrid seed maximises your seed or plant establishment, maximises grain yield, and also maximises the gross return to the growers. So, moving on to the winter types briefly. As I said before, we had three sites where we grew our winter type canola trials at Wallenbean in New South Wales, Inverleigh in Victoria, and Narracourt in South Australia. So the dry matter cuts were taken at the 6 to 8 leaf stage just prior to bud initiation, and grain yields were measured on both grazed and ungrazed plots. 3 generations were tested in these trials from the F1 fresh hybrid seed, retained F2 and retained F3 seed. So this is the grain yield and dry matter results. So for each of the different seeds, fresh F1 seed, retained F2, retained F3. Dry matter is in the blue lines. The orange bars are the grain yield for the one-grazed treatments, and the grey bars are the grain yields for ungrazed treatments. So looking at the grain yields, there's no significant difference between grain yield for the Hyola Feast, whether it was grazed or ungrazed. And the similar results occurred for both the retained F2 and retained F3 seed. But there was a significant yield decline by retaining seed. So it was a significant yield advantage to the F1 fresh seed. When you introduce grazing to it, to the equation, how does it affect stability of yield? So again, apologies people, I've only got one pointer I can use. So looking at the F1 fresh seed is in the blue, red and blue lines at the top. So the red line is your ungrazed plots, and at higher yield potential, the grain yield obviously increases. But grazing it does depress yield, and it has depressed yield in this set of data at a greater level at the higher yield sites. With the F2 retained seed, it's in the orange and green and purple bars. Green and purple bars. So green bar is this line here, and the purple one at the bottom. Similar result to the F1, that at higher yield potential sites, the grain yields of the ungrazed plots increased, with grazing depressing the grain yield slightly at higher yields. Finally, looking at the F3 retained seed is in the What do we... Sorry, I've got to just check that. Orange and brown line. So orange here and brown in the middle. So the ungrazed plots showed a similar response, that at high yield sites, the ungrazed plots increased, but slightly different reaction to grazing, in as much that grazing depressed yields at all sites at a similar level, similar rate. So, looking at the actual dry matter results in the blue bar, so there was a significant decrease in dry matter production between the F1 and the F2 or F3 retained seed for the winter canolas. And moving on from there, what does that combination mean for growers? So we did a similar simplified or gross value return by calculating how much the grain value was worth and attributing the seed costs to the grain value. The grain value returns were calculated by grain yield multiplied by grain cost, subtracting seed cost. The grazing value was estimated by using 60% of your dry matter yield multiplied by the consumption of 1.2 kilos of dry matter per head per day by lambs, with a growth rate of 100 grams per hectare per head per day, and a dollar value of $5 per kilo of live meat. So, because of the no significant difference in grain yield between the ungrazed and grazed plots within each seed source, grazing your winter canolas adds between $800 and $1,000 per hectare return to the grower. But because of the significant difference between the grain yield and dry matter yields of the F1, the total... pressing the wrong button again, sorry about that. The difference between F1 and retained yields, retained seed, we're looking at approximately $1,000 a hectare of extra value to the grower. So a quick summary on the winter canolas. F1 seed produces more grain and more dry matter than retained seed. F1 produces more grain yield than the retained seed, and grazing your winter canola will maximise your returns for the growers. So an overall summary for looking at both the winters and the springs is that F1 seed produces your higher plant establishment in the spring types. Your F1 produces the highest grain yields for both winters and springs. Your F1 seed produces the highest dry matter in the winter types. And your F1 hybrid seed maximises return on investment in most environments. And retaining seed will save you costs for your growers, but it'll also cost you more on profit. I'd like to acknowledge my fellow co-author, Willow Liddle, and Justin Kudnig, aka JK, or Captain Canola, as probably most of you would know him. I'd like to also acknowledge the work that the Pacific Seeds Canola R&D and the TD teams have put in, all the service providers who have helped with our trial management throughout the year, and to GRDC for the opportunity to present this data here today at Grower Update 24. Thank you very much. Okay. I'll leave it here so I don't walk off of it.
Which is what I did. Thank you, Andrew. And very challenging. Canola yields under half a tonne more profitable with F1. That's very challenging. A question over here. I think I recognise that face.
Mark Fowler, grain grower from Williams and Wickerpin. Andrew, the issue is not usually as a grower whether to grow F1 or F2. It's when you don't get your F1 seed that you have to decide whether to plant that or plant an open pollinated variety or no canola at all.
Yep.
So that's the important part of this analysis that I would suggest is missing, is the step down to an open pollinated.
Yep. Okay. Well, there is only a certain amount of work can be done each year, and research dollars are always limited and a scarce commodity. We certainly have a large set of data, other data that comes with these trials that we collected this particular set of comparative notes from, which compares a whole range of varieties from all of our competitors around the countryside, and you can get a fairly good idea as to where value comes in from hybrid versus OPs. And yeah, I think there is a yes, you're in a position where you have to make a decision if you cannot get your first choice of F1 hybrid, or you don't get enough. Yes, I don't have the figures off the top of my head to be able to give you the specific answer as to what yield potential do you swap from F1 to F2 or OPs.
Thanks, Andrew. About 80% of what we see in plant establishment is maternal. So it's the environment in which the seed is grown. So I'm wondering, how do you compare different seed lots that are grown and harvested from different environments and probably graded differently? How do you make that comparison when you've got different sources for where seed has been derived in comparing F1 self and then F2 self?
Well, you're going to have different seed sources for any hybrid seed wherever it's produced, because from all the seed companies have different production paddocks across the countryside, and that you can't produce different hybrids in the same paddock. So there's always going to be a complicating matter as to the actual seed source, and it's part and parcel of hybrid seed, unfortunately. But all the F or retained seed in these trials came from a single source. The retained seed was all graded to greater than 1.88 millimetre, whereas the F1 hybrids were as they came from production. So there would have been a range of seed sizes. We do have that data, but I don't have it. I can't remember off the top of my head to quote it to you. But yeah, there were some differences. So the way we pack our seed is the number of seeds per packet for the plot. So we effectively adjust our seeding rate to the seed size to target that 40 plants per square metre.
Yeah, David Hudson, SGA Solutions. In your analysis, it's all based on yield, but growers get paid on yield and oil content. Did you analyse the oil content of those trial results? And if so, what were the outcomes and impact on the data, both establishment and also gross margins?
Very, very good question. Yes, we have analysed them, but they were analysed post this paper being written. And so we do have that information, and we'll incorporate that now that it has become available. No, I haven't seen that data at this stage.
I think that's the bell.
I've been gonged.
Okay, thank you, Andrew.
Thank you very much.
And for those who want to ask you to put your hands together for Andrew, that's great.